Hey guys, this is Mr. Mahmood and you are watching Protein Synthesis Lecture 2. This is the second and final lecture for the Protein Synthesis unit, but again, just because it's only 2 lectures long does not take away from the immense importance of the information from these 2 lectures. This second lecture in particular is going to be a jumping point for a lot of the discussions that are going to be had throughout the remainder of the school year, specifically in terms of understanding how mutations affect organisms and affect all of the organisms that can develop in future generations, and how it can stimulate a whole lot of change that happens in terms of development and evolution and everything that comes from it. So it's very important you understand what a mutation is, as well as understanding how it can affect organisms. And then the final thing we'll talk about here is how we as scientists are using what we understand about mutations to actually start manipulating things ourselves. And this is what we consider genetic engineering, where we actually make mutations happen in our favor, or we force a mutation to happen. We control a certain change in the DNA for our favor to get an outcome that we're looking for, which we consider recombinant DNA. And then the opposite, if we're trying to eliminate any sort of a consistent change or eliminate the likelihood of a mutation, we'll talk about the process of cloning and how there's some particular patterns that I would expect you to be able to understand in that process as well. So this is all about mutations, this understanding in lecture, so please make sure you get it so that when we talk about this for the remainder of the year, you'll understand how the process works and therefore how it can truly affect all kinds of different organisms. So when I say the word mutation, I'm sure the first thing most of you think of is something negative, right? Some sort of deformity or some sort of disease and some major problem that would be a very bad thing for someone to have. But what's true about a mutation is that it's not always bad. In fact, as I hope you'll understand by the end of this, and as I hope you'll be able to connect as we go through the remainder of the school year, there are a lot of situations where a mutation is actually beneficial and helpful. And it's not as uncommon as you may think. Your cells have worked to become very efficient. We talk about DNA replication happening in the S phase of the cell cycle, and we're talking about 3 billion base pairs all getting paired up and matched up in order to make 2 copies of the original DNA line. That process is really efficient, but at the same time, mutations can happen, and they do happen. On average, every one of us has about 6 mutations in our DNA line. So we all have mutated DNA. Now, you can take that to mean something negative, or you can take it to mean something positive, but as hopefully you're going to be able to understand here, just because you have a mutation somewhere, by no means guarantees that that mutation is going to be represented in any kind of proteins that you're actually making. So there are some factors that are going to come to play in whether or not a mutation actually shows in terms of the proteins that are being produced and then the traits that those proteins represent. So before we get into that discussion, let's just go over a basic introduction of the general categories of mutations. I think the best way to try to visualize this would be to think of a general statement. So I'm going to put a sentence out here. I know there's no punctuation here, there's some grammar issues, I get that, but this statement just says, the sun was hot, but the old man did not get his hat. Now, as weird as that statement may be, there's a reason I chose it, and anybody who's watched the first lecture could probably tell me why I chose this particular statement. Hopefully you'll notice the 3-letter sequence for every single word. And if you remember, every codon of your messenger RNA that originated from the DNA is based off of these 3 base sequences that in turn attract a particular anticodon of tRNA and 1 particular amino acid. So I'm using a statement here that's broken down by 3s for a reason. Because if this was the original DNA line, as weird of a statement as this may be, it does make sense. The sun was hot, but the old man did not get his hat. It is what it is, right? But if I were to change just one part, just one letter, that statement can be significantly different. And there are 3 ways that you can just change the statement with a 1-letter change. One way is by switching the letter out for another, but the overall number of letters stays the same. So this is what we consider a point mutation. This is a comparison of a point or a substitution mutation, because the only difference here is in 1 letter. Everything else is exactly the same. So if you looked at this real fast, you probably wouldn't even notice a difference. But if you look carefully here, you'll read as it says, the sun was hat, but the old man did not get his hat. So there is a change here, but it all still kind of makes sense. Every word is legible. Every word is a word of the English language, and it's something that you can make sense of, even though there is a slight change that's going on here. The other two categories are what we call frameshift mutations. In this case, the one base change not only changes that one position, but it changes everything that comes after it. And so in this case, by adding a letter, it changed everything that came after it. So if you look carefully, everything seems normal until you hit that first T that follows the word hot. So the sun was hot is just like it was at the beginning, but now it looks like there's an extra T that was put in there, which means everything that follows now is going to get shifted one position to the right in terms of the three base sequence. So now every single word that follows no longer makes sense. It's not a word that's in the English language anymore, because what was supposed to be a first letter is now a second. What was supposed to be the second is now a third, and so on down the line. So everything gets shifted over. So not only are we dealing with a change that's going to happen with that one base that was put in, but now everything that follows is going to be messed up as well. This is what we consider an insertion mutation. We'll talk about that as well. And then this third category, again, is just a one-letter change, but it's going to affect everything that follows the change as well. In this case, a letter was taken out. And again, all the problems and all the changes are happening around that first word, hot, around the fourth word of the statement. In this case, the O of hot was actually deleted. So this represents a deletion mutation. And by taking out that letter, again, every letter that follows is going to have to adjust, this time to the left, in order to fill in that gap. So the third letter becomes the second, the first letter of the next word actually becomes the third letter of the previous, and so on down the line, again messing up every sequence that follows. So now we're not just talking about one word changed, but every word that follows after it. So you can see how these two frameshift mutations at the bottom are much more drastic, typically, of a change than what you see with that second line, or the example of a point mutation. So let's talk about each of these in a little detail. So again, a point mutation is only going to affect one particular letter. And because it only affects one particular letter, it doesn't change the number of letters overall, and therefore the three base sequences stay the same everywhere else. So as you can see here, the words before and after the spot where the substitution happened are not affected at all. You can see the sun was, those first three words are the same as they were in the original line, and then after the change from hot to hat, it's the same sequence that follows, but the old man did not get his hat. That sequence didn't change either. So now all of that part stayed as it is. The only area that was affected is that one particular letter, and then the one particular three base sequence that comes with it. That's why we consider it a point mutation. You can literally point to the one letter or the one base that's affected by the change. Nothing else is affected. We call this a substitution mutation because literally you're just switching one for the other, but you're not changing the overall number of bases that are found in that DNA line. A good reference and a perfect analogy is anybody who plays any kind of a team sport. So a lot of my students are into basketball, so we'll go with that. Basketball has 5 players on the court at any given time. And if I'm doing a substitution, I'm not changing the overall number of players. All I'm doing is switching out one player for the other. So the same number of players will be on the court at any given time. That's the same thing that's happening with a point or a substitution mutation. So it's really only going to affect one particular amino acid that would be attracted to it. Anything that came before the change or anything that comes after shouldn't be affected by the actual sequence itself. There are some slight exceptions to that rule, but that's the general idea, that everything before and after the mutation is not affected. Only the 3 base sequence is affected directly with the mutation. So in this case, if I had the original DNA line as CTT, so if I had CTT as the original DNA, you know that the messenger RNA line that would be read from that would be, in this case, GAA. If it was CTT, you should already be very comfortable with this. The messenger RNA codon would be GAA. But if the DNA were to have a mutation, in this case a substitution mutation, where instead of that second T, I now have a C in its place, so now it's gone from CTT to CTC, the mRNA codon that would have read from it has gone from the GAA originally to now having GAG. So if you look at the messenger RNA line and then you use that to read your codon charts, the GAA codon would have read for a glutamate or a glutamic acid. That would have been the original amino acid that was supposed to be matched up in that position. But you'll notice here, even with this mutation of the CTC in the DNA, which changed my mRNA line to GAG instead of GAA, if you look at the codon chart, what amino acid would it attract? The same glutamate. So you'll notice here that even if the mutation, if we're talking about something like a substitution, even if the mutation happens in a part of the DNA line where you're actually going to be reading and making a protein from it, it's still possible that you could still end up putting the exact same amino acid in the same position, which in this case would be true. So I'm still putting a glutamic acid or a glutamate in the exact same position, whether the DNA line is CTT or CTC, because as you'll notice with these codon charts, there are multiple scenarios where you can still end up matching for the same amino acid, even if you're using slightly different codon sequences. And usually that's the case if we're switching out the last base. So that usually the best chance of keeping the amino acid the same is if the third base is the one that's being switched out, because you'll notice that some of these, it doesn't really matter what that third base is, as long as the first and the second are what they are, it puts you in a very particular box where there's only one option. So that can happen quite often. A mutation can happen in the third base sequence of a codon, and it may not affect the protein at all because you're still attracting the same amino acid. So that's one example of how even a mutation that's read in a part that you're going to make proteins from, it still may have no effect. So this is where I tell you how the fact that you have 6 mutations in your DNA line on average doesn't necessarily mean you're going to show for 6 mutations in terms of all of your traits. First off, most of your DNA line is not actually red. Most of your DNA line is what's considered junk DNA, which means it's parts of the DNA that you don't actually use to make proteins. So if a mutation happens somewhere in there, it's not going to affect you. You're never going to make the proteins that would change because of that change in the base sequence. So the first thing with mutations is it may not even be in a position where you would use it to make a protein, so that mutation would never show up in any kind of a trait. The second thing would be, even if a mutation was represented in your DNA line that was actually a part that you're going to transcribe to turn around and make a protein, if it's a substitution like this, there is a possibility that it could still create the exact same amino acid position that it would have created without the mutation, which means the entire protein is exactly the same and would function no differently than it would have before. So here's another scenario where a mutation still could have absolutely no effect on the DNA line. However, just because a substitution mutation only affects one base does not mean it can't have a drastic effect on the overall protein. And here's a good example of that. Again, we're starting with the CTT original DNA line, which would attract GAA for the messenger RNA codon that would be transcribed from it, and then you attach that to a tRNA that would attract a glutamic acid or a glutamate. Now if I change it from CTT to CAT on the original DNA line, now my messenger RNA codon is going to read GUA. And if you look, GUA now codes for a valine. So instead of a glutamate or a glutamic acid in that position, a valine is going to be put in that spot instead. And that is a pretty significant difference. In fact, this is the exact substitution mutation that occurs in individuals with a specific disease called sickle cell anemia. Sickle cell anemia is a blood disease. It tends to be more commonly found in certain regions of Africa because, again, these are mutations. These are things that happen randomly in a genetic line, and it's been passed on so many times within a given region that certain parts of the world just happen to be more likely to show for some traits than others. So this is an example of a trait called sickle cell anemia that makes your red blood cells slightly change shape, which makes them sickle shaped. So instead of being the perfect round red blood cells that most of us have, they're oblong shaped and misshapen, which makes them very difficult for them to pass through your bloodstream, more likely to cause clots or blockages, and it also won't be able to carry as much oxygen as a healthy red blood cell would be able to. All of this change in your red blood cells happens just because of a single base change in the hemoglobin protein that's actually used to make these red blood cells. So just that single change, which is the exact same change you see here from CTT to CAT, the change from a glutamate to a valine, is enough to make that entire red blood cell function and look different. So this is an example of how a substitution mutation could absolutely have a significant effect. So just because it only affects one base doesn't suggest that it's something you shouldn't worry about or that it's something that shouldn't be able to affect you in any way. It's usually the one that's going to affect the overall protein less than the second category that we're going to get into. The second category is what we consider a frameshift mutation. With the frameshift mutation, again, like you saw with the example that you see up here, from the moment of the mutation, everything that follows is going to be completely skewed. And it's all because of the 3 base sequences that are going to be used to attract particular tRNA anticodons. So here's an example of a sequence of DNA bases. So you see, because I have 9 bases, I have 3 particular 3 base sequences that would in turn produce 3 mRNA codons and attract 3 particular amino acids. So the first 3 base sequence I have there is TAC. The messenger RNA codon that would be transcribed from that, I hope you guys know already, would be AUG. Alright, so AUG would be my first messenger RNA codon. The second codon, if I'm pulling from a GTA DNA line, would be what? CAU, good. And then my third mRNA sequence, if the DNA is ATC, would be UAG. Alright, so if you're using this as the example, look at your codon chart. AUG, if you look, would attract a methionine amino acid, which remember is a start codon. That start codon of AUG always attracts a methionine in that first place. So here's another example of how methionine is in the first position. In this case, I have CAU. If you look on the chart, you'll see that I'll attract to a histidine. That histidine is now in the second position, or it's the second amino acid in the protein. And then in this case, I have UAG, which if you look on the codon chart, is one of your 3 stop codons. There are 3 stop codons that you need to know in terms of the end of the protein line: UAA, UAG, and UGA. So in this case, UAG is one of those stop codons which would have ended this protein at that particular point. Now obviously, a protein is not just going to be 2 amino acids long, we're just using this as the example. But the third codon group that we had here would end the actual protein from being extended any further. This is what the normal DNA line would be. But if I introduce an insertion, the original DNA line, now I'm going to add a base. And I'm going to put an extra base right there after the first 3 base sequence. And the one that's underlined there is the base that's now been inserted. So because I put a new base in that position, everything that was originally following is now having to shift 1 position to the right. So because it's all getting to shift 1 position to the right, what was originally the first base of a sequence is now the second. What was originally the second is now a third. What was originally the third is now the first of the next 3 base sequence. So it's going to drastically change not only the 3 base sequence where the mutation occurred, but every 3 base sequence that followed, because literally you had to shift that frame of 3 bases over 1 position. So the first 3 base sequence, TAC, is exactly the same, as you can see, but this is where it starts to change. Now instead of GTA being the next DNA 3 base sequence, now it's C and then GT. And then the next 3 base sequence is now the A from the previous one plus the AT, which was the start of this one. So now I have AAT. And you'll see now that C is now sitting in that position off to the right, where it's now going to be the first base of that following sequence. And we are assuming there are still plenty of bases that follow, so now it would just be the first base of that new sequence. All of these, after the first 3 base sequence, are no longer what they were before. So in this case, going to messenger RNA codons that would be made from this line, originally the TAC wouldn't be changed, so I still would have an AUG, a messenger RNA codon be produced there. But in this case, now I have a CGT instead of the original GTA. So CGT, if you're following messenger RNA, would code for GCA. And then the third one we're going to follow here, now it's AAT, which is different from the ATC. AAT now reads for a UUA. And then that C that we're now looking at the beginning of the next sequence would be switched to a G plus whatever else would be that follows. So that next mRNA codon would start with a G when originally it could have started with something completely different. So now if you look at the actual amino acid codon chart and compare, you know again, because the first 3 base sequence stayed the same, the first mRNA codon is the same. So again, we're going to attract a methionine, which is again the start codon. So at least the protein is still starting in the same place, still starting with that methionine amino acid. Now the next sequence, GCA, is not the same as the CAU, so if you're looking it up on the codon chart, it's going to code for an alanine. So now I already have an amino acid that's not the same. But here's the most drastic problem. This is usually the biggest problem that comes with these 2 categories of frameshift mutations. It's not just the fact that we're changing some specific amino acids, but what was supposed to be the stop codon on the original line is probably not the stop codon anymore. What typically tends to happen is because of the shift, like you see here, instead of the UAG reading for a stop, which meant no more amino acids are made and that shut out the protein, now it's coding for another normal amino acid, in this case a leucine. So the sequence is now going to be continuously read. And that means more amino acids are going to add on than are supposed to until another random stop codon is reached later down the line. So that can make this protein significantly longer than it was supposed to be, which means it folds a lot differently and will make the protein function completely differently. Or vice versa, you might actually end up hitting something that reads for a stop codon before you were supposed to. And so now that protein is going to be way shorter. And again, you're going to have significantly different folding that happens, which makes the protein function drastically different as well. So let's compare this now to the other category frameshift mutation that we consider a deletion. Again, it's the same basic problem. We have the same 3-base sequence. We've talked about all of this already before. However, this time, instead of adding a base and shoving all the other bases to the right, I'm going to take a base out. In this case, I'm going to take out that first base of the second 3-base sequence of the DNA. So I'm going to take out that G that's in GTA. If I take out that G, now everything that was previously following it had to get shifted one position, this time to the left. So this time my frame is being shifted to the left because of a deletion, as compared to an insertion shifting it to the right. So this, again, the first 3-base sequence is the same, but now this sequence is changed because the G is no longer there, the G was deleted, and everything that followed had to be shifted one over to the left. And in this case, you see now it's TAA instead of GTA. In the 3rd-base sequence, it was ATC. Now it's just the T and the C, and then we just have to assume whatever was following is now shifted back as well. So all of these things are going to change, not just the position where the mutation happened, but everything that follows is going to change again. And this, again, can cause a drastic change. In the 1st-base sequence, the AUG stays the same. TAA now is the DNA line, is going to read for AUU. And then finally, TC, and we're assuming whatever that 3rd base might be, is going to now read for AG, and then whatever that 3rd base might be. So if you look on your codon chart, again, the first one's the same. You still have a methionine. Now AUU codes for a completely different amino acid, in this case an isoleucine. And now that A and G, depending on what the 3rd base is, will code for one of two things, either a serine or an arginine, as you can see from the chart there. So again, we have a serious problem. We have a couple of serious problems. We have different amino acids, so amino acids have changed, so the protein is no longer the same. But again, the most drastic problem here is what was originally your stop codon, and the position where the protein was supposed to stop no longer looks that way. So you understand what all these mutations are. Now let's talk again about the effect of these mutations. Like I said before, certain things have to be true if you want a mutation to have any effect at all for the organism. First of all, the mutation has to happen in a part of the DNA that's actually being transcribed to make proteins, which isn't all of the DNA. A significant portion of the DNA line is not being read at all. You only pull from certain parts of your DNA line to actually make the proteins that make you who you are. And every cell only pulls from certain parts. So there are parts of your DNA line that are never touched. And if a mutation is happening somewhere in that part, it's not going to affect you at all. Now, let's think about the different ways that it can show and have more drastic effects than others. When it happens in an organism's lifetime is very critical and very important, and in which type of cells the mutation may happen is also very critical and very important. So here's an idea. If I had a sequence of cells, I started with a cell, and that cell went through its replication process, made exact copies of the DNA and divided. Now I have 2 cells, and then these cells divide again, and I continue to make more and more cells. All of these are identical to each other because the process of mitosis and DNA replication is usually very consistent. So now let's say this represents almost the whole organism. This multicellular organism has gone through most of its growth and development with no mutations that are significant. But here towards the end of its development, let's say a significant mutation occurred. So now I have that cell that's significantly different than all of the ones before it. But we're to the point now where the majority of the cells that make up that organism have already been developed, which means we're pretty much to the point where almost all of the cells are functioning normally, and there are a fairly small amount that actually show for that given mutation. So if we assume these are the base cells that organism is going to have for the rest of their lives, those are the cells that are going to be replicating each other and growing. There aren't going to be that many mutated cells in that organism because they developed late in the development process. Now compare that to another scenario where this time, early on in the development, from that first cell, that zygote, somewhere in that first couple of rounds of division, the mutation occurred. So now if the mutation occurred this early on in development, as these cells continue to make more copies of themselves, now we're talking about a significantly larger portion of that organism containing these mutated cells. So if this is your starting point and this is the base organism, all of the cells that are going to be regenerated from those cells for the rest of that person's life or that organism's life are likely to show for that mutation. So the earlier a mutation happens in the developmental process of an organism's lifetime, the more likely those mutations are to show and have significant changes in the organism. And again, that's not always a bad thing, but it's going to be some kind of a change, and it's much more likely to show for that change if the mutation happens early. And it can't happen any earlier than in the very first cell of that organism. So that means if the mutation is something that's passed on to you by your parents, and so now your very first cell already has the mutation, so that means 100% of the cells that make up who you are have that mutation, then you're much more likely to show for that mutated trait than you would have been otherwise. And then if you think about passing it on to the next generation, it all depends on the type of cell where the mutations are occurring. If you have a mutation that happens in one of your body cells, one of those diploid cells that we've talked about before, which are things like blood cells, nerve cells, bone cells. Most of the cells of your body are considered diploid cells or body cells or somatic cells. If the mutation happens in one of those cells, it could very well affect you. But as you're going to learn in the next unit, in order for you to produce an offspring and for you to have a child, basically, you have to make gametes or your sex cells. For guys, we're talking about making sperm cells. For the ladies, we're talking about making your ovum or your egg. The only way that you can actually pass on a mutation to your offspring is if that mutation is able to get into the DNA of your gamete. Now, there are a lot of examples of how mutations can lead to particular diseases and disorders and disabilities, but as hopefully you're going to learn here for the rest of the year, if it weren't for mutations, we would all be bacteria. Every bit of life on this earth would be an anaerobic bacteria. There'd be no oxygen on this earth, there'd be no plants, there'd be no animals, there'd be nothing. We would all be bacteria if there were no mutations. But because of mutations, genetic lines change. And as genetic lines change over time, theoretically, if you follow evolutionary theory, new species can actually start to develop. Genetic lines can become so different from each other because of a slight mutation or a drastic mutation that organism now no longer can match up with another. So now it develops this entirely new species, and those species mutate as well and branch off. So one species can develop into another, and now we can get all this variety of life that's developed over the last 3.5 billion years because every generation sees slight mutations happening. So one population starts to change significantly from another until they develop entirely new species, entirely new categories of life. So we get that mutations affect organisms, and a lot of times it can have a negative effect. And we as scientists have sat there and watched this effect happen over and over again. And it's only been recently that we started to think, well, what if there was a way we can sort of make a mutation happen? What if we wanted something to be changed a little bit and we made it work? This is a process known as recombinant DNA, and a process of genetic engineering. Recombinant literally means to recombine. If you change the DNA sequence of an organism, then that part that's changed can potentially read for something completely different than what it was supposed to. Well, what if I wanted a cell to start doing something? And then what if I just took the DNA information that would do it, that would make that protein, and I just injected it and shoved it into an original DNA line? Now that cell is going to do what it normally did plus whatever I just added. And that's exactly what's going to happen here in terms of recombinant DNA. It's a basic process. You don't need to know an extensive explanation for it, but you need to be able to kind of follow what's going on here. In order to do this, it takes some basic steps. So I first need to isolate what's considered my gene of interest. And I'll go with a very common example here. It's something we just finished talking about a couple of units ago. Remember the feedback loop. If you have too much sugar in your bloodstream, your body secretes insulin from your pancreas. And we've also talked about how people who are diabetic can't make that insulin properly anymore. That means they have to get insulin, human insulin, injected into their bodies. Now, before this understanding of recombinant DNA, the only way that people who were diabetic were able to get insulin was to actually get it from either other species of animals who were close matches but not always perfect, or to try to harvest insulin from other human beings in multiple different ways. So you can imagine all of those had some significant risk factors involved in the process. But at that point, that's all they really were able to do. You need insulin, what else am I going to do? I'm going to give it to you somehow. This process has allowed human insulin to be produced in abundance in a laboratory with almost no risk to the patients who are getting the insulin injections. And they're doing it because they're taking the gene of interest, which is the production of human insulin, which is a protein, and making certain cells, in this case just random bacterial cells, start producing human insulin. So here's how it works. They take the gene of interest, which is whatever trait we're looking to make another cell do. In this case, the production of the human protein, insulin, pulled from your beta cells in your pancreas. So you find that gene of interest, the part of the DNA that actually makes that protein. You'll pull it out of the original DNA line. And usually the way it's pulled out is by making what are called sticky ends. That means it's being cut not straight down the middle of a full DNA line, but sort of cutting along the hydrogen bonds between the bases so that it makes for basically more surface area, so there's a better chance of reconnecting things when you're putting it all back together. So it's very specific restriction enzymes that are being put into play here. A restriction enzyme is the kind of enzyme that will actually cut DNA in very specific sequences. So if you're looking for one particular sequence, like anywhere you see a GTCA in the DNA line, the restriction enzyme will cut it right there. So anytime it sees it, it'll cut it at that position. So I have my gene of interest isolated out, just the segment I want. Then I'll cut another DNA line to put it together. Now, we use bacterial cells for a very particular reason for this. Bacterial cells actually have two different strands of DNA. They have one that's like ours, that's basically double helix rotated and it's kind of one long line that tends to be kind of crumpled in together. But they also have what's called a plasmid DNA. A plasmid DNA is a circular DNA that's found in bacterial cells. Now, let's think about why circular DNA would be beneficial here. If I need to open up DNA so that I can put this gene of interest back in its place, if the DNA was all linear, that means I would have to cut it open and have two completely separate pieces of DNA that I would then hope to stay close enough to themselves to where I can still glue both ends without losing either one of the original DNA lines. That's a problem. If I take something that's a long line and I cut it down the middle, the likelihood is at least one of those two pieces will never be brought back. They're going to be spread out and it'll be hard to bring them back together. But if I have a plasmid, which is a circle, all I have to do is cut one spot in the circle and open it up. If I can just keep it as a circular and just open up one part, and then take the gene of interest and just attach it in on both ends of that opening, now I have the whole DNA line together with the original DNA plus my gene of interest. So now we have that full plasmid, the recombinant DNA, is going to get re-injected back into the cell, and we're going to let that cell function normally. And the beauty of this is once you manipulate one cell, you let it go through the rest of its cell cycle. And part of that cell cycle is going to be its replication of its own DNA. So it's actually going to replicate all DNA that it has, including the stuff that you put in. So even the part that it doesn't actually have and doesn't actually make, it's going to replicate it because it's part of its DNA now. So that means if you take the time to manipulate this one bacterial cell and then just leave it alone, that cell will turn into two. And now you'll have two cells that have the full manipulation that you were going for. And those cells will turn into 4, into 8, into 16, 32. They'll go through their own binary fission process and replicate the full DNA that's in that cell, which includes your gene of interest. So by manipulating one bacterial cell into being able to start producing human insulin, now it can turn into millions of bacterial cells, all naturally producing human insulin, because they have that part of their DNA line. So just like they're making other proteins, they'll make that as well. And so doctors just harvest that insulin that's being produced in this laboratory in abundant amounts, and they'll turn around and put it to the pharmacies and give it out to anybody who needs it from that point on. And then finally, the exact opposite concept to forcing a mutation to happen is trying to avoid any kind of mutation whatsoever, and that's this process of cloning. The process of cloning, as you've heard the term before, is to create an exact replica of a particular type of cell or a particular type of organism if you're thinking on that higher level. But most of the time when we're dealing with cloning, we're dealing with what's considered therapeutic cloning for making specific types of cells that we can turn around and use for laboratory work or for particular treatments. It's rarely something that we're thinking about making a whole organism from, but the process is about the same all the way through. In terms of cloning, if I'm trying to make an exact replica of a particular cell, what you need to always remember is to follow the nucleus. Follow the DNA. If you can follow the DNA, you'll always see what the original cell and what the final cell are supposed to be. Because it's the DNA that is going to ultimately produce the proteins and make the traits for that organism. So if I'm looking to replicate an exact organism, I'm going to use one exact cell and its DNA. So what usually happens is you'll start with your cell that has the desired genes that you're looking for. So you're trying to copy a specific type of cell and make many of the same type of cell. You'll find that particular cell, and it's always a body cell or a somatic cell, and you'll take its nucleus out of the cell. You'll try to harvest and isolate the nucleus. So if you can pull the nucleus out and isolate it, that has the information that you want. That's the desired trait. That's the kind of cell that you're looking to make. So you pull the nucleus out of its cell. Now that DNA is going to have to go into a donor cell. But a donor cell has its own nucleus, but those aren't the characteristics and the traits you necessarily want. So you'll take the donor cell, take out that nucleus, and then replace it with your desired nucleus. So now by pulling the donor cell out, you put the desired nucleus in its place. So now you have a cell that's going to function based off of your desired nucleus that you want to have manipulated over and over again. And then usually the third step involved is going to be to take that cell and now put it into what's called a surrogate, or some sort of an organism that will allow that cell to go through its cycle and replicate over and over again, have all the resources it needs to make more and more of itself, until you finally end up with what's considered the clone, which is many of these cells that are all exactly the same as each other and exactly the same as that original desired trait that you were wanting from before. So let me give you an example. We'll deal with birds here. Let's say that I have a blue species of bird, and from that blue species of bird, I isolate its nucleus. I take the nucleus out of a blue bird. Then I take a red bird like this, or a cardinal in this case. I take the cell of that red bird. I take the nucleus out of that as well. And then I take the nucleus from the blue bird and put it into the red bird cell. So now I have a kind of a combined cell here that's the cell of a red bird with the nucleus of a blue bird. And then I inject that into a surrogate blackbird. So I put that cell that's a combination of red bird cell with a blue bird nucleus into a black bird to allow the cells to grow and develop into an offspring, into a baby, basically. And then that blackbird's going to lay an egg. What color bird do you think is going to crack out of that shell that blackbird laid? Is it going to be a blue bird, a red bird, a purple bird, some sort of combination with a black, or a pure black bird? Which bird is going to actually be produced through this process? The bird that would be produced from this process is a blue bird. So it's wherever the original DNA was. Always follow the DNA. Wherever the original DNA was that was put into that cell that's then going to start to grow, that's the exact kind of cell you're always going to make. There's no mixing, there's no combinations here. Just follow the DNA the whole way through. So this is an example of how you can try to eliminate kinds of mutations by just working off of one cell and trying to repeat that one cell over and over again. So you see how mutations can have a drastic effect on an organism. You see how that drastic effect is not always bad. And you also see how we as genetic engineers have actually figured out a way to force specific mutations to happen that will actually help us in the long run. So hopefully this made sense to you. And I guarantee you we're going to be talking about mutations over and over for the entire school year. So it's really, really critical that you understand how and why they work. Alright guys, take care. I'll see you next time.